Hello, biology superstars. Welcome to the content video for B1.1 about carbohydrates and lipids. And this is all common curriculum content. There's no extra HL information. So points 1.1.1 to 1.1.13 are fair game, whether you are in Bio SL or HL. Let's get started. So, since there's a lot of information in this content, I sort of split it into two sections, carbohydrates and lipids. The carbohydrate main ideas are carbohydrates are used to store energy. I'm gonna see if I can get my pen going. I'm gonna go purple. Yes. So carbohydrates are used to store energy. There are many different types of carbohydrates. We'll go over some specific ones the IB expects you to be familiar with. Monomers of carbohydrates are called monosaccharides. Polymers of carbohydrates are created from their monomers in condensation reactions. We'll go over condensation reactions a little bit. Polymers are broken down back into monomers in hydrolysis reactions. We'll go over the basics for those two chemical reactions. And important carbohydrates specifically include different forms of glucose, as well as starch, glycogen, cellulose, and molecules called glycoproteins, which are very important in cell-to-cell recognition and on the outsides of cells. All right, so first point of information, chemical properties of a carbon atom allowing for the formation of diverse compounds upon which life is based. So here we're sort of just looking at the basic information about carbon as an atomic element. Students should understand the nature of a covalent bond. I'm going to get my pen again. Covalent bond. And you can go back to theme A if you need more information about this. We talked about these already in theme A. And that a carbon atom can form up to 4 single bonds or a combination of single and double bonds with other carbon atoms or atoms of other non-metallic elements. Carbon is non-metallic as well. Include among the diversity of carbon compounds examples of molecules with branched and unbranched chains and single or multiple rings. So we'll look at some of those as examples. A nature of science statement is that students should understand the scientific conventions that are based on international agreement. So the metric unit prefixes kilo, centi, milli, micro, and nano. And I don't really feel like the IB does such a good job of talking about this anywhere else. So when you're in SI metric units, you start with a base unit. So like, for example, let's say our base unit is a meter. And then you add prefixes that decide the way in which you're talking about that unit. So with the base unit of meter, the prefix kilo would be 10 to the third, and that's always true. Like a kilo anything is 10 to the third. A centi, which it would be 10 to the negative 2, so then you're getting smaller than a meter. Like if you can picture on a ruler, a lot of rulers are demarked in centimeters. A milli would be like 1,000th, which is 10 to the negative 3. A micro, I think I'm going to go over here. A micro would be 10 to the negative 6. And a nano would be 10 to the negative 9. And I would say that the IB does sort of expect you to know these, like to know these specific prefixes. They're named in the syllabus. So I would go ahead and memorize these. And sorry, that is so messy. All right. So talking more just about the basics of carbon, compounds that contain carbon are generally called organic, and carbon compounds are present in carbohydrates, lipids, nucleic acids, and proteins. Form and function is very important in carbon molecules, and as the statement of information said, the IB wants you to understand like some sort of examples of both branched and what do they say? Branched and unbranched, and also single and multiple rings. So some good examples are, which we'll just draw in here. Back to the old pen. So an example of like a sort of unbranched basic molecule would be start with carbon in the middle. It's connected to hydrogens all around. And remember, it can bond 4 places. This last place is also a carbon. This double line represents a double bond. And then this OH we've talked about before represents a hydroxyl group. And this molecule overall, which I don't really think you need to know what it's called, but this is acetic acid. A good example of a ring structure is like glucose. So we'll draw as a ring starting with an oxygen. And every place that you see like a kink, like a turn like this in the ring, you can assume that a carbon is there, even if it's not drawn in. So like this is a carbon, 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 and this is a carbon for 1, 2, 3, 4, 5, 6 total carbons in a glucose molecule. Also, anytime that you don't see, like if there's a hydroxyl group here, you draw it in. If you don't see that drawn in, you can assume that it's a hydrogen atom, a lone hydrogen atom. You'd like tend to not draw those in all the time in molecular structure drawings. All right, so those are some of the sort of shapes that I think IB would expect you to be familiar with. It does seem like if you're taking the exams 2028 or later, they're including some of those in the data booklet. So I don't really know what else they would want you to know about it. Next point of information. Production of macromolecules by condensation reactions that link monomers to form a polymer. So here's where we're going to go over these condensation reactions. And also examples of polysaccharides, so big sugars, polypeptides, so proteins, and nucleic acids. Obviously, in this section, we're going to really focus on the polysaccharides, on the carbohydrates. Polypeptides and nucleic acids sort of get their own sections. But we are going to talk about this condensation reaction. So let's get started with that. I'm going to go forward one slide. Boom. All right, so condensation reactions create larger product molecules from smaller reactant molecules. So for example, if you start with 2 monomers, this is a monomer and a monomer of some type of a carbon-based molecule, the way that they will generally condense is there are a lot of hydrogen and hydroxyl groups hanging out on these molecules, as we saw in the last picture. These will, I'm going to change colors, these will combine with each other to form a water molecule, and you will be left with the 2 purple circles that represent, in this case, our carbohydrate molecule joined together, and then let's go red. Let's go red, red, red. I think that's orange, but whatever. And then a water molecule off to the side, separate from the 2 carbon-based molecules. And you call this, I'm actually going to change the color. What has just happened is a condensation reaction. And so, for example, you could have a molecule of alpha glucose and a molecule of beta glucose, so 2 different isomers of glucose. We'll talk about that a little bit more in a future point of information. Linking together to form maltose, which is a disaccharide. You don't need to know so much about like the difference between disaccharides and polysaccharides, but remember that prefix di means 2. So this means 2 monosaccharides linked together. The bond that links them together is called a glycosidic bond, and chains of only beta glucose would form cellulose instead. We'll talk about all that in another point of information. All right. I think that's pretty good for condensation reactions. Let's move on to hydrolysis reactions. So the next point of information, B1.1.3, is about digestion of polymers into monomers by hydrolysis reactions. This picture is the same because these reactions can go both ways. So if you are building up a bigger molecule, that is a condensation reaction. If you are breaking apart a big molecule to create smaller molecules, that is a hydrolysis reaction. So in hydrolysis reactions, water molecules are split to provide the hydrogen ion and the hydroxyl groups, or hydroxyl group, that are then incorporated to produce the monomers, hence the name of this type of reaction. Water, lysis, breaking apart of. So you are breaking apart a big molecule by adding water to it. It's right there in the name. So in a hydrolysis reaction, you have the polymer as the reactant. So I think mine was purple. So you have the big molecule with the water off to the side. But in this case, the water is going to get added in to that big molecule, and it's going to break apart into two smaller monomer molecules, one with an OH group and one with a hydrogen ion. And the process that has just happened here, I forget what color I used for this. Was this that pretty blue color? Not that color. That's good enough. All right. This is hydrolysis. Oh, it's nice how it's blue because it's like watercolored. All right, and if you understand those two types of chemical reactions, you're in pretty good shape. The next thing the IB syllabus looks at is the form and function of monosaccharides. Remember, monosaccharides are the monomers of the carbohydrates. And students should be able to recognize pentoses and hexoses as monosaccharides from molecular diagrams, showing them in ring forms. Remember, this prefix pento means 5 and hex means 6. So we'll go over what the ring forms of 5 and 6 carbon organic molecules look like in 1 second. Use glucose as an example of the link between properties of monosaccharide and how it is used. So this is that thing of like linking form and function. And the functions or the properties that you should be familiar with are solubility, transportability, chemical stability, and the yield of energy from oxidation as properties. But first, let's just quickly go over the idea of 1, 2, 3, 4, 5, 6. This is a hexose sugar because it has 6 sides. Glucose is an example of that. This is a pentose sugar because it has, I might have said 5 sides for the hexose. This has 6 sides. A pentose sugar has 5 sides. And ribose, the sugar that we saw in nucleic acids, is an example of that. And we'll talk a little bit more about glucose specifically. So it is the most common monosaccharide in nature. It is 6-sided. It is a hexose ring, and it has 2 important isomers, which are alpha and beta. And I would say that the IB doesn't really expect you to recognize the difference between those anymore. So I might draw it in just because it feels sad not to, but I don't think you really need to know very much about it. All right, so I'm going to just get my pen going. So oxygen molecule. Remember, every time there is a turn in this shape, you can assume that it is a carbon molecule, including one carbon molecule coming off the side of the ring. So this is C1, C2, C3, C4, C5, and C6. If, I'm just going to do it because I can't not do it. If it is alpha glucose, the hydroxyl group is down here. So this is alpha, and that means there's a loose hydrogen up here. This is at the first carbon position. If it is beta glucose, the hydroxyl group and the lone hydrogen switch places. So I'm going to like erase this as much as I can. Go back to my pen. This is beta, and that means that the lone hydrogen is down here. And I remember this like beta has its hand up and is waving bye-bye, bye-bye, bye-bye beta, because its hydroxyl group is up here. All right. Next up about glucose, no matter what isomer you're talking about, is the properties that the IB expects you to know. There are basically three of them. It is a soluble molecule because it is polar. It can dissolve in water and be transported in blood. It's stable because it has that ring shape with all of those hydroxyl groups situated in the axial regions of the molecule, like around the outside. And this stability is important in all of these polysaccharides that chains of glucose form. And it can be oxidized. So oxidation is addition of oxygen to a molecule. Really, usually you think of oxidation in terms of electrons. So I remember either, I'm going to go with my actual pen, Leo Ger or oil rig. I like oil rig better. Oxidation is loss of electrons. Oxidation is loss of electrons, and reduction is gain. And we're not going to go any deeper into oxidation and reduction in this particular content. If you're in the HL class, I'll talk about it a little bit more when we talk about cellular respiration and photosynthesis, because they are like important reactions in that. But you can see this happening here. From here to here is reduction. From here to here is oxidation. All right. Let's go here. Good. Okay. This. Go. All right. Polysaccharides as energy storage compounds include the compact nature of starch in plants and glycogen in animals due to coiling and branching during polymerization. The relative insolubility of these compounds due to large molecular size and the relative ease of adding or removing alpha glucose, and we know what alpha glucose is now, alpha glucose monomers by condensation or hydrolysis to build or mobilize energy stores. So you can see good examples of form and function here. These form and function. Both of these molecules are polysaccharides composed of repeating chains of monosaccharides, but because of the composition of alpha and beta glucose, even though they're both like composed of glucose, they end up looking and functioning very differently. All right, so starch is the primary polysaccharide that is used in plants. It is linked to alpha glucose molecules. It has two forms, amylose, which is here, that is like a straight line, and amylopectin, which is here, that is branched. Glycogen is excessive glucose in animals. It's similar to branch start, but there's even more branching. So you can see in the glycogen there's like so much branching. And cellulose is the form that's in plants. So the structure of cellulose related to its function as a structural polysaccharide in plants is the point of information here. And because of the formation of this molecule, and we'll talk about alternating orientation of beta glucose monomers, you have this straight chain molecule that can be grouped in bundles and cross-linked with hydrogen bonds. So when you look at cellulose, this is like what cell walls in plants are made out of. And you can see here, and I got the pen just a little bit, the position of each of these monomers of glucose is different. They're like at 180-degree angles to each other. You can tell because of this 6 carbon and also the position of this oxygen atom in the ring. So like this is carbon 1 going from over here. This is 1, 2, 3, 4, 5, and then hanging off the side is 6. So you have here 1, 2, 3, 4. So this is a 1-4 linkage that makes a straight line across. I think the easiest way to think about starch and glycogen and cellulose is to compare them all to each other. So the next table, this table does just that. You should be familiar with all the information on this table. So like where is cellulose versus starch versus glycogen, like where is it found, what is the source tissue of it usually, what are the subunits of it, because remember they're all glucose but it's different isomers of glucose, what are the bonds that form in it, does it branch or not, what is the diagrammatic structure of it, like you're not going to be asked to draw this, but it's helpful to maybe be familiar with it. And I think this is a good thing to just like pause the video and study this table for a few minutes. All right. Then we'll talk about, I think, our last point of information for polysaccharides, which is the role of glycoproteins in cell-cell recognition. And we are going to talk specifically about ABO antigens as an example. So here you can see a picture of a glycoprotein. It's like an integral protein with a little glycogen chain hanging off the top of it, protein with a carbohydrate attached. And let's go forward one slide, talk about this just a little more. So glycoproteins have a protein part and a carbohydrate part. If you were going to draw one, which can sometimes be helpful, I would draw like the protein part like that and the carbohydrate part like that. Protein, carb. And since we're looking at carbohydrates, the carbohydrate part is like the defining part of this molecule. They're very important in cellular membranes because they are used in cell-to-cell communication and recognition. And a really good example of this is in your blood cells. You know how people say you have like type A or type O or type B blood? That has to do with the glycoproteins that are present on your red blood cells. So I'm going to get my pen going. So like let's say you have A blood. A blood means that your blood has glycoproteins that are a certain shape on it. So like this would be what, I mean, not really, but like this could be a representation of what your blood cell looked like. B type blood, you would have a different glycoprotein on the outside of your red blood cells. If you have AB blood, that means that you have both of those. So AB blood is both the circles, or like whatever, what we are representing as circles, and the squares. And if you have type O blood, it means that you do not have any of those marker proteins on the outside of your red blood cells. You have like a little place where they could be, but you don't have them. So this is important because your blood attacks blood cells that are foreign to your body. So if you get like a blood transfusion or an organ transplant that has the wrong type of blood still present in it, which they would never do at the actual hospital, but whatever, your blood attacks blood cells where it doesn't recognize these chains on the outside of the proteins. So that's a really important example of glycoproteins and cell-to-cell recognition. All right, we're going to keep going to the lipids information. If you need a little break, this is a good time to pause. Lipids, main ideas. Both carbohydrates and lipids are used to store energy. There are many different kinds of lipids, the same as there are many different kinds of carbohydrates. Monomers of lipids are glycerol and fatty acids. We'll talk about how they join together. The health properties of lipids depend on the structure of the fatty acids they contain. This is important, and we'll talk about saturated and unsaturated fatty acid chains. And steroids are lipids that are used as signaling hormones in the body. So steroids are like what you also call lipid-based hormones, and they are sent from an organ that secretes hormones to a target tissue and do some type of signaling in the human body and in plants and stuff. Like lots of different living things have steroid hormones in them. All right. So the first thing to understand about lipids is that they are hydrophobic. They are water-fearing. Lipids are substances in living organisms that dissolve in non-polar solvents, but are only sparingly soluble in aqueous solvents. They include fats, oils, waxes, and steroids. Let's, I think, just go right to the next thing. So fats, oils, waxes, and steroids, a little bit more information here. They are all non-polar. They're oftentimes referred to as hydrophobic because they do not dissolve in water. As a note, oils are generally fats that are liquid at room temperature. I'm going to see if I can get my pen. Hello, pen. I'm liking this blue. All right, oils, liquid at room temp. Some oils have enough saturated fat that they are solid at room temperature, and we will talk more about saturated versus unsaturated fat in the next point of information. So for here, we'll say mostly. All right, and let's get to this thing of saturated and unsaturated fatty acid tails. That is important when you're talking about the formation of triglycerides and phospholipids by condensation reactions. So we already talked about condensation reactions for carbohydrates. The general components of the reaction are the same, but when you're talking about lipids, you have one glycerol molecule linking either three fatty acid molecules, and that is to form a triglyceride, 3 plus a glycerol, or two fatty acid molecules and one phosphate group. And if there's a phosphate group, you call it a phospholipid. And I think the easiest way to think about this is to do a little example. So a triglyceride, for example, is this glycerol molecule. Excuse me. This this glycerol molecule. No, I'm backwards. Yes, this glycerol molecule plus fatty acid tails. And the R groups here are like the number of molecules that are in the fatty acid tail. So these could go for a long time, boop, boop, boop. And when these two components come together, it's a condensation reaction because they release water. I think that's all I was going to say about this. Yes. And so you have, triglyceride, glycerol, 3 fatty acid tails. For a phospholipid, you have a glycerol still. but it is linked now with one phosphate group and only two fatty acid tails. These are the types of lipids that you see forming a bilayer in cell membranes. And generally, the fatty acid tails are facing in the middle, and the glycerol heads are facing the outside. So like that. So that might look familiar from like intro bio or something. And this idea of the difference between saturated, monosaturated, and polyunsaturated fatty acids is really important in those tails. So include the number of double carbon bonds and how this affects melting point. Relate this to the prevalence of different types of fatty acids and oils and fats used for energy storage in plants and endotherms, respectively. So let's do a little definition of the saturated versus the unsaturated. If, I'm going to get my pen going, if a fatty acid tail is saturated, that means that hydrogen is bonded to the carbon at every possible position. The carbon is saturated with hydrogen and only single bonds exist within a fatty acid tail. If you have an unsaturated, so this is saturated. If you have an unsaturated fatty acid tail, that means that there are places where the carbon is double bonded to another carbon, and that creates like little kinks in the tail. It also means that one side of oh no, I'm drawing oxygens. It also means that one side of the carbon is not bonded to a hydrogen on its other side. So like, because the carbon can bond 4 times, 1, 2, 3, 4, 1, 2, 3, 4. So you'll notice there's no hydrogen here and no hydrogen here. I'm gonna erase because it's like a little confusing looking. I wonder if I can make the eraser bigger. I guess not. So this idea that an unsaturated fatty acid tail turns because its form is not to be fully saturated with hydrogen, and that affects the way that it looks. It also affects its function and things like its melting point. Unsaturated fatty acid tails have a lower melting point and are more likely to be liquids at room temperature. And you can also talk about cis versus trans unsaturated fatty acids. Cis is when the lone hydrogens are on the same side. That's like the one we just drew, and that's when the molecule bends. Trans unsaturated fatty acids are lab produced, and what they can do in a lab is move. I'm gonna actually go. So this and this would be like the normal organization for this tail, but you can do something to straighten it out. And what you do is transfer or move one of those hydrogen atoms to the other side. And this would be what's making this a trans fat or a trans unsaturated fatty acid tail. And this is helpful in food production because it creates very shelf-stable food molecules and products. But they are actually not considered very healthy, and industrial trans fats are currently banned even in the United States. But you should be able to recognize, I think, both the cis and trans positions for those fatty acid tails. Next point of information is about triglycerides in adipose tissues for energy storage and thermal insulation. Students should understand that the properties of triglycerides make them suited to long-term energy storage functions. Students should be able to relate the use of triglycerides as thermal insulators to body temperature and habitat. So here is a lot of information that sort of does all of this for you. This has to do with long-term versus short-term energy storage, how much calories of energy each of these store in them. And it has to do with their thermal insulation properties based on their ability for heat conduction. So this table is another one I just like stop and look at it for a few minutes and make sure all that makes sense and maybe think about memorizing it. And when you, just as a definition that I think is important, when you talk about adipose tissue, that is like a fat tissue, is tissue where triglycerides are stored in animals beneath the skin and around some organs. There's actually technically 3 types of adipose tissue. I'll go my pen. 3 types, which are white, which is like what you commonly think of as fat tissue, brown, and beige. So we are talking about the white one here. All right. And then we move on to formation of phospholipid bilayers as a consequence of the hydrophobic and hydrophilic regions of a phospholipid molecule. And you should be familiar with this vocabulary word amphipathic. So we'll go over that a little bit. I guess I'm actually going to draw this right here. Okay, so here are the phospholipids. So like here's one little side of a cell membrane. Here is the other side. So you can see phosphate group, bloop, 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 with fatty acid tails. This is a phospholipid bilayer. Phospholipid bilayer. All right. And this word amphipathic so this is Greek, comes from Greek. Amphi means both, or amphis, like an amphibian or an amphitheater, where you can see both sides of the stage. The amphitheater wasn't like necessarily immediately obvious to me. So that is from the Greek root amphis, which means both. And then pathic comes from pathera, which means feeling or suffering. Pathera, so like an amphipathic molecule can be both hydrophilic feeling and hydrophobic feeling. Okay. All right. And just some general information about phospholipids. The hydrophilic part of the molecule is what is attracted to water. That is the phosphate head. The hydrophobic part is what is repelled by water. Those are the fatty acid tails. And really, if you were going to like draw this super correctly, which not very many people do, there would be a glycerol molecule and then a phosphate head and then 2 fatty acid tails. And these are found in cell membranes, which we talked about already. All right, and the last, I think, point of information, last point of information is the ability of non-polar steroids to pass through the phospholipid bilayer. So this has to do with fats because steroids are lipid hormones. We will talk a little bit about estradiol and testosterone as examples. Both of these you probably recognize as like what people commonly refer to as sex hormones. Signaling molecules that decide primary and secondary sexual characteristics, and they are both similar in shape, and students should be able to identify compounds as steroids from molecular diagrams. This does come up again, just FYI, in C2.1. about chemical signaling. So especially if you're in HL, we'll talk about this more when we hit theme C and level of organization 2. Chemical signaling. But these molecules, so these are testosterone and estradiol molecules. Here is, I guess I would say that this is specifically estrogen, but whatever. It is in the oestradiol family, and testosterone. All right. And you can tell the structure of these molecules because steroids have, oh, look, they are labeled here for you. Steroids have 4 fused rings. So this is cortisol, not estrogen, but whatever, just note that. Cortisol is another example of a steroid hormone. And they are nonpolar, so they are lipid soluble, not water soluble, which means they can travel directly into your cells. They pass through the cell membrane and initiate a cascade reaction. They're carried in the blood because they don't dissolve in the blood, and then they enter directly into your cells. So I think that's it for B1.1. Yeah, that's all she wrote. All right. See you in the next lesson.